Thank you all for being here today on this kind of rainy day, but it's a good time to talk about science. So it's always a good time to talk about science because we like science and we like math, and we especially like genetics, and that's what we're going to talk about today. So as you heard from that wonderful introduction from Teresa, thank you, I am heading the Center for Genetic Medicine, and it's an incredibly exciting time in human genetics, largely driven by changes in technology, but really translating to being able to help and influence how we practice medicine right now. So we'll start with just a little bit of primer about the genome and the complexities of human genetics, but it all comes from the fact that the genome is a really big place, the human genome. It's 3 billion bases are what we consider the length of all the genes lined up together, or all the DNA sequences lined up together. And of that 3 billion bases, 1% of it specifies protein, and 99% of it does a variety of things, including regulating a lot of the expression of other genes. And so all of it matters, but there's been a lot of attention focused on the 1% that specifies the proteins. And if we divide it up into genes, there's approximately 20,000 genes. I've had people ask me, exactly how many genes are there? Well, the answer is sometimes genes are embedded in genes, and so it's an approximate number. And this is a picture I actually grabbed off the website, and I like it because it shows the old-fashioned way we used to do sequencing. So here you see a scientist looking at what are lanes of sequences, and that's what I called lane-at-a-time sequencing, meaning that if you looked at this, and this is how I grew up as a graduate student many years ago, being able to look at 200 or 300 of the 3 billion bases at a time. So you get the idea, it would take a long time to look at all of them. And what transformed human genetics was about 10 years ago was this idea that we no longer do lane-at-a-time sequencing, but instead sequencing is now done in an array format using fluorescent tags most of the time. And so by doing it in an array rather than in a lane, we can do millions of bases at the same time. And so therefore, it now becomes feasible to actually really sequence not only the first human genome, as was completed around the year 2000, but lots of people's genomes to be able to learn more. And so array-based sequencing is really what next generation sequencing refers to. And because it's all done in parallel, sometimes people will also call this massively parallel sequencing. So with that, we now, for the first time, have available databases full of lots of people's sequences. And so for the first time, we get the idea of actually being able to understand human genetic variation. And the short answer is humans are a lot more variable, different from each other, than really we ever could have imagined. And this is from a review written by Jim Lupski a couple of years ago, and I like it because, first of all, it highlights the fact that we are, in fact, all related to each other. We are one giant family. But it emphasizes the idea that when we consider variation in humans, that there are different types of variation, and the biggest difference is how frequent a variant is in the population. So what there is there's a lot of common variation. So if I look at a particular position in the genome, it's 50% of the people might have an A in that position, 50% might have G. We call that a high frequency variant, meaning it's very common in the population. But most of the time, that high frequency variation is actually quite ancestrally old. And so what you see is that's the very old variation that's been there many, many, many generations ago, and it's been somewhat fixed in the population. And then we fast forward to thinking about what is considered evolutionarily newer generation, or rare variation, meaning that if I look at a position in the genome across 100 or 1,000 people, I might find only one person carrying that particular variant. And the reality is most of the variation that exists in each of us as humans, about 85% of our genetic variation, is actually rare. And only about 15% of it is common. So the answer is we're a lot more diverse from one another, and each of us is really quite unique. And the people that we are most closely related to, our immediate family members, there we share some of our rare variation. But as we get a little bit further and further, it really just becomes that 15% of the genome that's more common, high-frequency variation that we share with others. So the rare variation is an area in which we, from a research perspective, have a lot of interest in being able to look at that. So this is another way of thinking about that same idea, which is if we look across a population, and we imagine these diamonds here are each individuals, and we're looking at common variation, this high-frequency variation, this has actually been the study of things called genome-wide association studies, where we associate this particular variant with the development of hypertension or with the development of any common disease you might think of. And there have been lots and lots of these genome-wide association studies, but because they involve very large populations, the overall effect on the outcome is usually a very small effect, high across the population, but small from an individual perspective. And we contrast that to thinking about rare variation, which I'll fast forward again into thinking of in the rare diseases. We actually now know the function of lots and lots of human genes because they've been associated with rare diseases. And in fact, when we think about the whole population, about 10% of the population as a whole is affected by a rare disease. And that's because we have all this rare variation. So if we look across the population, there might be only 1 in 100 people or 1 in 10,000 people that have that disease. But within that family, as that gene runs in the family, that genetic effect is very, very strong and very, very meaningful for their outcome. So the most common examples of these are some of the earliest single gene or Mendelian disorders ever solved were the breast cancer genes, BRCA1 and BRCA2. These genes really pave the path for being able to do what we do in the rest of genetics right now. And I'll talk briefly about one called LQT syndrome or long QT syndrome. That's also a genetic disorder that was one of the earliest ones solved. So these are the disorders for which we're now seeing ever expanding genetic testing becoming more and more available to people. And with this increase in genetic testing, we're starting to appreciate that there again is a lot more variation than what we ever imagined. So long QT syndrome, I'm a cardiologist, so I like talking about cardiology things. Long QT syndrome is something we measure off an EKG. The EKG specifies the beat in the heart, and if you actually measure the distance going from this deflection to this deflection, this is actually the QT interval. And a longer QT interval is actually associated with pathology and can be associated with irregular rhythms, and in the worst case scenario, actually sudden death. And we know that this is due to rare variation in a subset of genes that code ion channels, which regulate really the EKG of the heart. So what's happened now is that we know that there are multiple different genes and many, many, many different genetic variants that can lead to long QT syndrome. So if we're going to order a genetic test, we see a family that has long QT syndrome, we want to do a genetic test, we now order what's known as a panel of genes. So that in this case, if we ordered it from this company, which is called GeneDx, we might get this panel of 15 genes. Other companies might have a few more or a few less on their panel. And what they do is that next generation sequencing across all of these genes in order to look for that rare variation. And here's an example. This is a 24-year-old woman who came to us because it turns out most of her life she had had a lot of fainting episodes. And at some point, somebody did an EKG on her and measured that interval as we go from the Q to the T, and it turned out to be quite long. And so based on that, we went and ordered a genetic test on her, and she had a mutation in one of the genes. This is an arginine substituting a cysteine, or R174 cis, we call it, and that's the diagnosis of long QT type 1. It was actually the first gene listed on, these aren't listed by that, but here's LQT1 is listed on here, and that's her cause of disease. So what's interesting is she is symptomatic. It actually turns out this is relatively easy to treat. In her case, we treat her with beta blockers, and in fact, she's now pregnant with her first child, so we'll have that available for following her children. But it also leads us to say, well, what about the other members of your family? So we screen the other members of the family. We identify those individuals in the family who carry the same gene. In this case, it's a dominant disorder, so it's about 50% of the family members carry it. But not everybody expresses it the same way. So most of the time, it's actually a mild disease, and only in some severe cases do people need, for example, to have defibrillators placed. And so in this case, it allows us to observe and continue to follow those people who need a little more attention from their cardiologist versus the people in the family who test negative for having this mutation. Then we're allowed to say, you guys don't have it, you didn't get the gene, off you go, have a good life, you don't need to worry about this, and none of your children that you have one day need to worry about this either. So that's the real value of being able to do genetic testing. But what we've learned through a number of years of doing lots of genetic testing for many different disorders, whether we're talking about cardiology disorders or cancer disorders, is that mutations express themselves in very different ways, even within the same family. And so this is an example of a family that I've taken care of for a long time. This individual came to me, he was about 65 years old at the time, and he had a genetic mutation that caused his heart muscle to be weak. And at the same time he was showing up, His daughter was showing up right after her second pregnancy, where she was having profound heart failure symptoms, and we knew right away what it was. It was related to this same genetic defect. And she was having symptoms much earlier in life, and likely the stress of pregnancy was actually bringing it on for her. Now, she's actually done really well. The nice thing about these disorders is they respond beautifully to medications. She's done very well, and in fact, just called us up again last week saying, it's time for me to come back into the doctor. So she's just needs to be followed from time to time and maintain taking her medicines. And so with this, we see that there are individuals in the family who show up at a much younger age, some at an older age, and we're now beginning to understand that the things that change the way the mutation expresses itself have to do with a variety of other factors, some of which mean there are other genetic variants that they carry, and some of that is environmental factors. And so when we do genetic testing, that variant that we may find in an individual that we consider pathogenic as likely causing a disease is really just a marker. It says this is an individual we want to follow more closely. It doesn't necessarily mean they're absolutely going to show up with disease. And so we think that's a really important point to always emphasize when we do genetic counseling. So that's also depicted here, which is we very often will find what's called a primary pathogenic variant, and whether I was talking about cancer or cardiac disease, we'd consider this the same way. There's often a single driver mutation, which is accounting for most of what happens, but then we know that there are things that modify the outcome. And sometimes those secondary modifiers are other genetic variants, which may be common, high-frequency variants, or maybe rare variants, or they may be other things like other diseases. So for example, if you have a cardiomyopathy mutation and you happen to have high blood pressure, you're probably going to show up with disease in your heart a little bit earlier in life, and that's the reason and the impetus for treating that blood pressure very well. Interestingly, for a lot of the cardiomyopathies, we know that being male actually makes it worse. And we don't know whether that's just the simple fact that males have bigger hearts and therefore wall stress is a little bit more on their heart, or whether it's something much more complicated than that. We know exercise, very intense, high-level, competitive, athletic exercise can make hearts worse for these individuals who run in these families. Now, that doesn't mean we tell people to go be couch potatoes. We instead just maybe come up with more cross-training activities, less repetitive exercise, and usually not very high-level, intensely competitive activity. And of course, other cardiovascular disease, or if we were talking about cancer, other things would stimulate that. We know diet is probably a big modifier, and we're only at the very beginning in being able to understand how diet and genes interact with each other. So how do we think about human variation? Well, I alluded a little bit earlier to the idea that we're starting to have large public databases of human genetic information. And one of the first available databases actually came out about 5 years ago. It was called the 1,000 Genomes Project. These were initially 1,000 individuals who were recruited to have their entire genome sequenced at low coverage, and this is all available in a public database. And the way this got off the ground was it was an international effort, so there were 200 people from the United States, 200 from Europe, 200 from Africa, and it also was a first ethnically diverse sample of human genomes, and again, started to give us this idea that humans are a lot more variable when we consider across the population. This has now been updated and actually contains about 2,500 genomes, and the difference of this database is it actually does cover the entire genome. There are several other large databases which have focused on just that protein coding region of the genome, the 1% of the genome, and there's now a database of about 65,000 individuals that we routinely use. And again, very, very diverse database that helps us consider variation. But all variation, some of it's benign, some of it's pathogenic, and so now we've started to consider each individual variant of how do we want to score it. So they range from being pathogenic or disease-causing. Sometimes we get tests back and it's called likely pathogenic, meaning the testing company or the researchers who've studied it say, we think this may be causing a problem, but our level of evidence isn't good enough to really say that this is pathogenic. And then we have many, many variants that we consider benign. And in the world of rare diseases, the biggest thing that makes something benign is the idea that it's high frequency in the population. We're talking about rare diseases, so the variants must be rare. And then we often get testing results back that are called variants of uncertain significance or variants of unknown significance. And there what we do is we will work with families and test that variant within their family and try to make the difference of understanding whether it goes with what the phenotype is so that we know whether it's pathogenic or not. And very frequently when we work with families, we actually can tip that balance one way or the other in considering those variants. But the biggest thing that helps us understand these variants is those large publicly accessible databases of human genetic variation. So now when we order genetic tests, they come back and they say, well, we found it in 0.1% of the genomes in the EXACT database, or it's been reported in these 3 families. And that type of information that's publicly available is what's helping us interpret these genetic variants. There's also ways of looking at the human genome. Again, these are publicly accessible tools, all our tax dollars working hard for us. The UCSC, which is the University of California in Santa Cruz Genome Browser, and I was hoping it would be up because today they were actually supposed to be unveiling their new version of it. But essentially, in this genome browser, you can look up any gene. And so here I've looked up BRCA1, which we're showing here, and this is the gene structure. And in that, it will actually list all the commonly described variants associated with that gene. And if you scroll further and further down, you can actually learn all about the epigenetics that are across this and what the different histone marks are around it. So this is a phenomenal genome browser to be able to look at genes, and it's a great place to just sort of play around and see what's going on. Another really important database that we use all the time right now is something called the Database of Clinical Variation, or ClinVar, as those of us in the genetics world refer to it. This is actually all the variants that have ever been described in humans. And as you can imagine, this database is rapidly growing to where it can barely contain all this information, but it is still nonetheless usually pretty useful for being able to look up information. So again, in this case, I'm looking up BRCA1 and we're looking at particular variants, and in this case, whoever put this in, so it'll say here it was reviewed by an expert panel and this one is considered benign. And this type of information where individuals from testing companies or research laboratories can all put their data into one place is what is making it possible to now be able to understand human variation. So it's an amazing collective when you think about it, that just sort of organically grown up and become the powerhouse of being able to interpret genetic variation. So just a couple words about cancer genetics, because again, cancer genetics really did lead a lot of the way for human genetics. So we distinguish cancer genetics into sort of 2 main flavors, which is germline mutations. Those that are passed through the germline means that it's inherited within the families. And the ones that we do a lot of testing for include breast and ovarian cancer, colon cancer, and then others with diminishing frequency. These are the big ones for which there is real genetic testing. And I think most women know that if you have family members that have had breast cancer, those individuals really should be tested for BRCA and the other breast cancer mutations. Similarly, colon cancer, especially early onset, multiple members of the family, those individuals should be tested. Because if you then find that mutation, and you can check that mutation in other family members, you know who in the family needs to be observed much more closely, just like we talked about in the cardiac setting. And that distinguishes from somatic mutations. And somatic mutations are the mutations that happened in the tumors themselves. And this is, again, an incredibly growing area where we want to be able to take tumors, rapidly and reliably profile them so that we understand what are the mutations that have happened there, and then be able to target our therapies against the cancer based on what genetic mutations that tumor has acquired. Now, what happens is tumors are pretty complicated. Tumors are different from the host. If I even sample a tumor at 3 different places, depending on the type of tumor, I may find different genetics in different parts of the tumor. And when I follow that tumor over time, as it's responded to different therapy, it's also changing its genetics. And so what happens in the tumors is vastly much more complex than what's happening in each of our germline genomes. But nonetheless, this information is incredibly powerful for guiding therapy, but it's just a more complicated place to be doing things. But there's great progress being made in this right now. And part of the struggles with doing somatic mutations is that very often it turns out there are big chromosomal rearrangements that are happening in the tumor. And as I talked about the sequencing, what we're doing with the sequencing is little short reads of sequence. When we do that parallel sequencing, we might be reading 150 base pairs at a time. And if the chromosomes all rearranged itself, little short read sequencing is particularly challenging to try to see bbig chromosomal arrangements with. So this is from 2013. I think many people saw this when it first came out. I know I was very interested by this. This was Angelina Jolie's op-ed piece that was in the New York Times when she did BRCA testing. So both her mother and I believe her grandmother before her and her aunt all suffered from ovarian cancer. And based on that, genetic testing was done and a BRCA1 mutation was identified. Angelina Jolie herself very publicly underwent this testing, was identified to carry this same mutation, which placed her at 87% risk, lifetime risk of developing breast cancer and a 50% lifetime risk of developing ovarian cancer. And based on that, she decided the choice that was right for her was to go forward with a double mastectomy and reconstructive surgery. Now, that's not the choice for everyone, but the point of this article, which I think she's done actually a very good job making this case, which is she had a choice. To know this information is to actually arm yourself with information and put yourself in a good position to be able to make what is the best choice for you. This is her follow-up, which came out last year, which is she then went ahead and actually underwent oophorectomy as well because, of course, her mother had died of ovarian cancer, and that's what she wanted to really protect herself against, and so she's made those decisions. This is probably the strongest decision an individual can make to undergo these surgeries. Many people will just choose to undergo continued monitoring. So one of the things that has, again, breast cancer has led the way in so much genetics, and one of the most important things was that genetic testing for breast cancer for many years was offered only by one company, Myriad Genetics. And what Myriad Genetics was able to build over that time was a very good database of information to help be able to identify what's a pathogenic versus a non-pathogenic mutation. Now with public databases coming up, we're now in a position that that testing can be offered by more than Myriad Genetics. And so one of the companies that's coming online right now, and the goal of many of these companies is to make genetic testing affordable and widely accessible to the population. So what Color is doing is they've put together a panel of 30 different genes, including BRCA1 and 2, and they're offering this testing for $250. Again, making it widely accessible to individuals, even when they're facing the idea that their insurance may not cover this. And we can talk in a minute about genetic testing and whether insurance covers that. So this company is really, really pushing the frontier of dropping the cost of genetic testing. And what's interesting is that this testing is usually very easy to do. It frequently now can be done off a spit kit, so you don't even need to have your blood drawn. You can literally have the kit sent to your house, do a spit, send it off, get your testing back. Notably, this company uses both physicians. The tests are actually ordered by physicians, and the results are all delivered by genetic counselors, who are critically important in this part of it. So what's happening is that I told you it was right around this inflection point in the curve, which is that massively parallel sequencing came into being. And with that, the cost of genome sequencing dropped dramatically. So the first human genome, which was done around the year 2000, cost $3 billion and took 15 years or so to get done, whereas now we can sequence a genome for pretty darn close to $1,000. It's about $1,100 to get a genome done right now. But while we're generating that huge amount of information, what hasn't kept pace is our computing skills, and that's what this actually shows. This here is Moore's law, which states that the timing at which processing speed improves on our computers. And while our phones have all gotten pretty smart, our desktop computers haven't gotten that much faster over the last 10 years or so. And so what's happening is we now can generate sequence at a much faster rate than we can begin to analyze it. And so we're seeing this backlog of sequencing developing because the computing can't keep up with it right now. So I say this because this was from 2009. This is actually the first whole human genome we did in my laboratory. It was actually a family who had lost a son at the age of 17, and they were interested to know what the genetic variants were. And they looked into doing some genetic testing, which at the time would have probably cost about $25,000 to do a small handful of genes, whereas Illumina, even at the time, the company Illumina, was offering a whole human genome for less money than that. So they actually elected to do entire human genome sequencing, and very quickly they were sent this hard drive, and this terabyte drive contained the human genome, and even just trying to analyze that genome was proving near impossible to do. So we actually they did a first pass analysis at Illumina. This is what it looks like. But when we first received this data, I mean, it essentially would shut down any computing cluster that we had where we tried to upload and analyze the data. And so from that, we actually developed some different technologies, and I'll talk about that in a second. But when you look at a human genome, how much information are you really looking at? So I keep saying it's about 3 billion bases. This sequence was not quite, at that time, 2009, we weren't getting quite 3 billion bases. We were getting about 93% of it. And in this case, that was about 3.3 million unique, what they called unique variants at the time, or 3.3 variants identified. And of those, totally novel variants were 150,000. Now, if we updated this now, those numbers would look a little bit different. In fact, we would find that if we sequence any one person's genome here, they would have about 4 million differences from the referent genome. So 4 million differences per individual. And of those, again, about 85% of them are really rare, most of them not even reported in public databases. So it's a huge amount of information for even one individual genome. So why is it so much information? Because we're doing this short read sequencing. So every single base, and again, 3 billion bases, say we're reading it at 100 length, that means we have about 1 billion data points that we have to align to the 3 billion bases of the genome. That is computationally very, very intensive to be able to do. To handle that amount of terabyte of data, that's a huge amount of information. And so many people have decided, well, let's not sequence the whole genome. Since computationally we can't quite handle that, let's just only look at that 1% that makes protein. And that's called doing an exome. And these days you can actually order an exome as a clinical test. That can be done as a clinical test. And it is much easier to do. It's only 1% of the genome. It's a lot less information. It does focus on the parts we know best. But it's probably not as good as doing a whole genome. We know the whole genome covers things better, but computationally it's a lot harder to do. So what do I mean by differences in data? So if I were to order one of those gene panels, like the Color company is now offering for BRCA1, that would probably be about not even a gigabyte, a tenth of a gigabyte of data. Whereas if I were to do an exome of data, the 1% of the genome, and cover it at good depth to be able to figure everything out, that's about 6.2 gigabytes of information. Whereas if I'm going to do a genome and I want to cover it at about 30 to 40-fold, cover each base that many times, that's more than 100 gigabytes of data, and it's a lot of little pieces of data. So that's a lot to look at. So when we first started doing this several years ago, we actually reached out to our colleagues at the Argonne National Laboratories because we're lucky. They're here, they're local, and they have one of the computers known as Beagle, which is a Cray XE6 supercomputer, and it is actually outfitted for biological purposes, which in computer speak means it actually knows Java, which a lot of the language we use was actually written in Java at the time. Now it's using different language. But we worked to take all the abilities to look at genomes that had been developed by the Broad Institute at the Whitehead, and we made them work in the supercomputing environment. So what does that really mean? That means that here's the number of genomes. If we were to look at 240 genomes, and we were going to try to do that on my desktop computer, it would take about 50 years to be able to do that. And I'm impatient. I'm not going to wait 50 years for that. If I were to do it in a small computing cluster, that still would take a couple of years to be able to do it. But on the supercomputer, we can do that in 2 days. And the reason why is because a supercomputer is built to move large amounts of data very seamlessly in almost 0 time. And so that's one way of being able to look at genomes. It is available to us, and we can do it that way, but we're actually now working with high-performance computing here at Northwestern to be able to get a bunch of nodes set up so that we'll be able to do that here. So let's talk about genetic testing. Who should get genetic testing? So as I mentioned, there's about 10% of the population with rare diseases. By and large, those are almost all genetic diseases. And so if you have and you know that something like that runs in your family, that's a good family to think about genetic testing. Also in the pediatric setting, when we see young children who have disorders, that's a good place to think about genetic testing. And importantly, when we're working with the adult onset or later onset disorders, or even the young ones, we want to test the person in the family who we suspect has the disease, because we want to be able to identify what is the genetic tag that marks that disorder. What type of genetic testing should we consider? Well, lots of times our first pass these days when we do genetic testing is we do panels of genes. If we're doing breast cancer, we don't just do BRCA1 and BRCA2, we do a whole panel of genes. If we're testing for cardiomyopathy, our panels are now close to 100 genes that we're doing at the same time. And so you can question, well, maybe we should just jump to exome. If we're doing that many genes, maybe we should just be doing exome. These days, and this could be different by next year, but right now we usually do exome testing for, again, a lot of the pediatric disorders where we really don't think our gene is likely to be covered on the panels, and so we will do that. And then we're reserving right now genome testing for those who've had clinical genetic testing where we haven't found anything, and in the research setting we're pursuing genome testing. And when should we think about genetic testing? Well, I think you really want to do genetic testing when it's going to clarify the diagnosis, which is almost always it's going to put clarity on the diagnosis. It's going to provide important prognostic information, and or it's going to direct your treatment options. And we can say that for all of cardiovascular and all of cancer genetic testing right now. We're seeing it now increasingly make its way into neurogenetic testing and many different movement disorders and epilepsy, where it will clearly guide treatment as we go forward. So we can see that the indications for genetic testing are starting to go up and up and up. And for every individual that we see that we do genetic testing, we're then also connected to all of their family members. So it's a clearly exponentially growing area for us. So what about genetic testing and the law? Well, the important part is to distinguish between when we do genetic studies in the laboratory, which is research, versus that being done by what we call a CLIA-certified laboratory. When we send genetic testing to a company or we're doing it in an academic laboratory that's been CLIA-certified, it must be done in a CLIA laboratory in order to be used in healthcare decisions. So if we intend to use that information in the care of that patient, we're going to put it in the medical record, it needs to be done in a CLIA-certified laboratory. So there's a clear distinction between clinical testing and research testing. They are not the same thing. A second question, and probably the question we get asked most of the time, which is, can I be discriminated against based on my genetics? And a number of years ago, it was actually President Bush that signed the Genetic Non-Discrimination Act, signed that into law, and it's called GINA, says that you cannot be discriminated against for health insurance or your employment based on your genetic testing results. So if you are found to carry a genetic variant and that places you at risk for a disease, you cannot be denied a job because of it, and you cannot be denied health insurance because of it. Now, important part about GINA is GINA doesn't protect an individual from disability insurance or from life insurance. And so when we're doing genetic testing on individuals, we will often say, go sign up for your life insurance before these results come back. Because at that point, it's a completely honest, good faith, you're signing up for life insurance. People who have life insurance know they may sometimes ask about your family history. Well, that's kind of like asking about your genetics. And so that they can make those decisions. So you're not protected for life or disability insurance, but you are protected for your health insurance and your employment. And GINA is actually the law of the land. Second question, and this goes back to that Myriad discussion, this came up a number of years ago, was the idea of, can genes be patented? And for many years, genes were actually patented. The BRCA1/2 tests were actually patented by Myriad, and that's why Myriad was the only place offering that testing. And so it went to the Supreme Court, and the Supreme Court essentially said you can't patent a gene. So that's what made genetic testing then spring up in a number of other places being able to offer the testing for that. And it was indeed a case that went forward based on BRCA1, a woman who wanted to have testing, wanted to get a second opinion on it, and there was no laboratory in the land that could offer a second opinion test on that because no one else could do the testing. So what the law did say is that the gene couldn't be patented, but that information about BRCA1 variability, which ones are pathogenic and which ones are not, that was in Myriad's database, that was still protected under the law. That was their work product, and they were allowed to keep that. But now with public databases being what they are, the power of that database is now much more public. So none of the genes that were in early days were patented. That was all the long QT genes, a number of genes, dystrophin, and then all the BRCA1s. Those patents no longer hold. 23andMe. 23andMe is a company that offers direct-to-consumer testing. Direct-to-consumer means that they weren't involving a healthcare provider in doing that. So you could get your spit kit at home, do your spit, send it off. Here they were ordering a little special of $199. You could get this done. And they were a little bit vague about what it was they were doing with your DNA sample. So what they actually do with 23andMe is they do the common variants, that about 15% of the variation of all the high frequency variants that are in the population and what those associated with. And then of course a select few rare variants are on what is their microarray chip that they use. And that's why they can offer this for less than $1,000. They actually got told to stop doing this for a little while because the FDA decided that what they were doing constituted providing medical care and they weren't medical providers. And now they've come back online in this last year because now they've got genetic counselors and they do have medical providers involved in it. So what they can do is give you lots of cool information about ancestry. So if you're really interested in your ancestry and want to find long lost relatives, this is an awesome way of doing that because remember, those common variants are very old in the population, so you'll find you have lots of relatives. And it can provide a little bit of information about genetics, but it isn't the same as genetic testing for rare diseases. They do offer some nice opportunities for research because what they do have right now is a database of probably close to 600,000 individuals that have participated in this, and they do have a lot of genetic information, and they do partner with research in academic institutions for those who are interested, but they don't have full sequence information. It's really just the SNP approach. So with that, you're hearing things like lots of publicly available information, your genetics, your risk, all sorts of kind of scary sounding things. Of course, everybody should be saying, well, what about privacy? What about genetic privacy? Well, one of the things that comes up, and this may change now from, a year from now, this may be a different answer. But one of the questions that comes up is if I have that terabyte of information of somebody's genome, is that actually identifiable? If it doesn't have the person's name on it anywhere, is that truly an identifiable piece of information? Because HIPAA, which protects all our privacy, just says, can it be reasonably identified? And really, a sequence as it exists isn't readily identifiable. Now, as databases build, and if you build a database and you put people's names and you allow a link between names and their genetic information, then yes, people do become identifiable. But as it exists right now, that doesn't exist anywhere. So it would require a large database of names linked to DNA sequences. And the only places I know where that exists right now are the FBI has one, and that's full of lots of criminals. So I think that they're going to protect that one okay. And 23andMe has a pretty large database right now. But again, they have got everything coded, so it's not like it's easy to get between the DNA sequences. But that could change over time. There are some people that believe that any stretch of DNA sequence, that if it has variation in it, is potentially unidentifiable and may be able to identify you. But right now, practically speaking, they're not really identifiable, but that could change in the future. So a couple of last words on reproductive considerations. What do people do with genetic information? So I work with all sorts of individuals and families that have genetic disease. And one of the things we talk to them a lot about is what their reproductive choices are because of knowing this information. And the answer is, for some people, they have a certain genetic disease or they've seen members of their family with it, and it's powerfully important to them that they not pass that on to their children. And so for those individuals who really don't want to pass on their genetic disease because let's say it's a really terrible genetic disease, early onset, all the features that make it a bad genetic disease, there are a lot of options available for people these days. And one of the ones that we have several people who are successfully doing this right now is pre-implantation genetic diagnosis. That's the idea that if you know the mutation in the family and that couple really doesn't want to pass that information on, they can actually do a genetic test by peeling off one of the cells of the developing embryo and doing a genetic test on that, and then only choosing to implant an embryo that doesn't carry the mutation in the family. Now, you get quickly, I'm talking about something that then involves in vitro fertilization, so it is much more complex to do this. But for some individuals with some diseases, this is really the only way they'll consider having a family is if they have this option to not pass it on. But, and I stole this off someone's website, but the idea of being able to peel off one cell and do a genetic test, if that seems hard, yeah, that's really hard. And you want to make sure that you're doing that accurately so that you really can make it possible that those individuals don't pass that on. But we've got a couple families right now that are midway through pregnancies doing this, and we've taken a few people through it over the years. So we like when it can happen and work out well for them. So last couple word on what else do we do with this genetic information and what else are we thinking about in the future? Well, we know that lots of genetic information is very important for telling us how our bodies respond to different drugs. We talked about it in the context of cancer, but we know that just regular drugs that you might take for high blood pressure or for managing your lipids respond very differently depending what your genetic predisposition is. And so there's a big area of growing interest in pharmacogenetics, of being able to understand how genetic variants change the way we metabolize drugs. And I think we'll see that being something that comes very readily into the medical record in the next couple of years. Many people are actually doing this testing right now, but there seems to be a gap of how do we take that information and put it into Epic so that we can see that answer and know how to respond to that. So there's some challenges there. And then in the world of rare diseases, or sometimes less rare diseases, we're also seeing gene-specific treatments being developed. So I work with children who have muscular dystrophy, and we actually had a big group of us testifying in front of the FDA last week about developing these antisense compounds that allow skipping around the mutation that these children have and allowing them to actually correct their underlying gene defect. Now, right now, the therapy is still sort of in its infancy, and it isn't as potent as it needs to be yet, so we've still got some steps to get through. But it does look like it's working in these early days. And then the one we hear a lot about in the press these days, about 3 years ago, there were new methods devised called CRISPR-Cas9 for being able to go in and site-specifically change a genome. And so the first efforts of this were actually done by a Chinese group in a developing embryo, and that raised a lot of ethical questions around the world right now about should we actually be doing that. The scientific community thinks, appropriately, we're not ready for that. We can't do it safely, we can't do it accurately enough, and what are the ethical implications of doing that? And so this is a large area of discussion right now. Very useful in the research setting, being able to make cell lines and being able to do things that basic researchers can do, but I think this is pressing a lot of individuals to think very carefully about, do we want to change the human genome in a way that it could be changed for future generations? So the genetic reality is we're a mix of a lot more variation than ever was imagined. Some of that variation is common variation. Some of that variation is rare variation. It's powerfully poised to predict different healthcare outcomes for us and for us to be able to take that information and make good decisions. So this is, I'm really showing this picture to highlight this woman right here, who is Lisa Castillo, the genetic counselor that I've worked with for many years. I can't do what I do without the wonderful genetic counselors that we have all throughout the system here. So we have a really great group of genetic counselors between Lurie and Prentice and FSM and Northwestern Medicine, and they're really tremendous professionals. We're growing our genetic counseling program here at Northwestern. We have one of the best, if not the best, genetic counseling program in the country, and we're expanding now, so we'll be taking 20 students this next year because we know this is a growing area. And so with that, I will stop, and I'm happy to take any questions.